Hello, and welcome to the webinar. I'm Kate Osterly, Marketing Specialist for Genomenon, and I'll be your host today. Today, Eva Sarah of Congenica and Mark Keel of Genomenon will be presenting real-world examples of efficiency gains in the analysis and interpretation of genomic data. They have a lot of great information to share, so I'll get right to housekeeping and introductions. As you are watching, please feel free to submit questions through the chat window on the right. Eva and Mark will answer the questions after the presentation. And so without further ado, I'll introduce our speakers. Mark completed his MD PhD in Molecular Genetic Pathology Fellowship at the University of Michigan, where his research focused on stem cell biology, genomic profiling of the hematopoietic malignancies, and clinical bioinformatics. He is the founder and CSO of Genomenon, where he supervises the scientific direction of the MASTERMIND suite of software tools. As part of the Congenica clinical team, Eva contributed to the analysis of rare disease patients in the Genomics England 100,000 Genomes project and is actively working on clinical interpretation of patient data from other projects. As a product manager, Eva drives the integration of new product features into Congenica's platform. So Mark will begin now with a brief overview of MasterMind, followed by Eva, who will spend the majority of our time demonstrating Congenica. Mark, take it away.
Thank you, Kate, and thank you everybody for attending the webinar. We're very excited about this partnership between Genomenon and Congenica, and as Kate suggested, my role will be limited here just to expose those who are unfamiliar with the MasterMind software and search engine, what it is, and how it can be used, and then I'll pass it back over to my colleagues at Congenica to continue with the webinar. So I'll begin by stating the problem that we're addressing. And that is to say that manual literature curation is extremely labor intensive and requires a great deal of expertise and doesn't scale. And part of this problem is access to the information that's available one at a time in the medical literature. And the problem that MasterMind has solved is by pre-aggregating, organizing, and annotating that information to the tune of 30 million titles and abstracts, fully indexed on a nightly basis, and more than 7 million completely indexed full-text genomic articles, including many hundreds of thousands of supplemental data sets, looking for the mention of any one of the diseases in the whole spectrum of human disease, any gene in the entire human genome, and any one of the variants in any one of those genes mentioned anywhere in any one of those articles or data sets, no matter how an author may describe any of those terms. And I'm excited to announce that within the past month, we've released a 2.0 version of MasterMind that streamlines a lot of the search capability and has paved the way for the addition of additional data streams, including the human phenotype ontology data set, which allows users to search for and return results that have to do with genetic phenotypes. I'll begin by saying that MasterMind is cloud-based for your genomic variant search and allows you to search through the entirety of the medical literature using any one of those indexed entities that I described, diseases, phenotypes, genes, or variants, and we will soon be adding drugs to that canon of our potential inputs to allow our clinical users and research users to be afforded the most expedient window into the content that's relevant to their clinical diagnosis for genomic information or otherwise research activities determining disease gene or disease variant associations. This is a window into what the MasterMind search result page looks like. I'll draw your attention to the left here where you can see for any given gene that you search, the entire comprehensive variant landscape for that gene, represented at the top as a Manhattan or lollipop diagram along the linear axis of the protein, with each bar representing an individual variant, and the height of those peaks representing how often the variant is described in the literature. And then all of that information is provided to you in list form below and can be used to refine your search to enhance the specificity of your results. To further enhance the specificity of your results, if you have a variant that you've identified in a dozen or more articles, if you'd like to extract or prioritize content that's specifically germane to a particular clinical question, such as befits searching according to ACMG variant interpretation guidelines or AMP or ASCO criteria in the context of oncology searches, MasterMind has a large battery of canned key terms organized in this case along those categories of evidence to inform ACMG clinical interpretation, but will also allow users to perform free text searches to, again, in a very flexible way, enhance the specificity of their search results. On the right side of this result page, I'll draw your attention to the literature landscape for the inputs that you've provided. The most typical use case, and the use case that Eva at Congenica will be describing when she talks about the integration of MasterMind into Congenica software, is a variant level search. So if as a user your question is, which articles have described my variant, what are they, and what information can I glean from them? That's what you're seeing here on the right in the form of the literature landscape for your search result. And just as I described on the left with the visual graphic display of that landscape and then the list form below, the top feature on the right here is the literature landscape with each icon representing an individual article, the x-axis being a reflection of the publication date of the article, the y-axis being a reflection of the citation score or impact factor for that article, and then the size of each of those icons, which represent an individual article, being a reflection of the relevance of the content of those articles to your individual search term. And then again, all of that information is presented for you to search, sort, and filter through in list form below. So again, I'll remind you that MasterMind is a database of genomic information built largely on the evidence in the empirical medical literature, comprising disease phenotype, gene, and variant indexing, decorated with many hundreds of pre-canned clinical and functionally significant key terms, as well as allowing our users the capability of performing free text search to further enhance their results and their use case in MasterMind. So with that background set for MasterMind, I'll pass the control back over to Eva, who's going to guide you through the result set and the use case for Congenica software. Eva, take it away.
Thank you very much, Mark. Thank you very much all for joining us. It is a pleasure to be presenting to you all today. So as Kate mentioned at the start, the title of this presentation will be real-world examples of efficiency gains in the analysis and interpretation of genomic data. And specifically, I'll be talking to you about the challenges that we currently face in the analysis of NGS data, and then present some real clinical cases where workflow efficiency was possible thanks to the use of Congenica's clinical decision support platform. So in terms of the outline for the webinar, I'll start by telling you a little bit more about who Congenica is and what is our mission. Then I'll discuss some recent findings that uncovered the through time and hidden cost implications of NGS data analysis. And then we'll see how we can maximize efficiency, diagnostic yield, and confidence using Congenica's clinical decision support platform. And lastly, I'll be sharing with you 3 examples of efficiency gains in real-life clinical cases. And I'll be doing this in a live demo of each case, demonstrating Congenica. So who is Congenica and what do we do? So Congenica is a digital health company that is helping healthcare professionals and organizations to rapidly process and interpret NGS data and convert it into actionable answers. So we have pretty much a very clinical heritage, being founded from partners from the Sanger Institute and also Great Ormond Street Hospital in London. And we are a span out from the DDD study, so Deciphering Developmental Disorders, which I'm sure many of you are familiar with. So the DDD study was led by Dr. Matt Hurls at Sanger and also by the UK National Health System, or the NHS. So Congenica has a very global presence. Our headquarters are in the Wellcome Genome campus in Cambridge, UK, where the Sanger is also based, but we do have international presence globally, including US and China. We are specializing particularly in rare disease cases, and especially those hard to solve complex cases. So perhaps some of you are already aware, Congenica has developed a clinical decision support platform that is really helping clinicians and clinical scientists and researchers to rapidly process NGS data in the form of gene panels, exomes, but also full whole genomes. So Congenica's platform has been demonstrated to improve diagnostic yield, helping users to get at the genetic answer much faster than traditional methods. It has also been demonstrated to maximize workflow efficiency from sequencer to report, thanks to intelligent automation. And finally, it has been shown to increase diagnostic confidence. And this is all thanks to a consistent, standardized, and reproducible workflow that helps you to improve every diagnosis. So although we are a relatively young company, we really have a global user base, and we are very proud to support a lot of partners throughout the world. We collaborate and have customers that are diagnostic laboratories, national genomic programs, and many other flavors of research centers. And in this slide, you can see some of our collaborators and customers. Of particular note, I would just mention Genomics England. Our engagement with Genomics England started as part of the 100,000 Genome Project in the context of rare diseases, but it's now extending to the full rollout of the National Genomic Medicine Service as part of the NHS, where whole genomes will be analyzed using Congenica. So I'll now pass to the second section of this presentation, which looks at the pain points, true time and cost implications of NGS data analysis, and particularly in the case of rare diseases. So since the advent of NGS sequencing, there has been this widespread expectation that the $1,000 genome will soon become available. However, we can all appreciate that this price only really reflects the consumables component and does not account for all of the steps that are part of the sequencing pathway, which really extend far beyond just the sequencing itself. This was highlighted in a recent paper that was published in Genetics in Medicine, and I've put the link to the paper just down below in that note. And this paper, the authors were trying to uncover the true time and complete costs of routine clinical grade sequencing in the context of a UK NHS accredited laboratory. So this lab was running around 400 samples per year, including rare disease and cancer. And for this webinar, I'm just going to be focusing on their rare disease findings. So to give you a little bit more context into what these authors were looking at in terms of their case collection, so they were looking at pediatric and adult cases, mostly trios, that had a broad disease spectrum. Importantly, similarly to what many other labs conduct, they were running a two-stage bioinformatics analysis. So first, they would be running a clinical analysis, trying to solve patient diagnoses, looking at just known genes for each disorder. And then if nothing was found, if the case was negative, they were running a research, let's say, gene agnostic exploratory analysis, trying to find other possible genetic causes for each case. In terms of cost data, the authors pretty much looked at all of the costs of the sequencing pathway, and particularly looking at interpretation and reporting and accounting for staff times and staff salary and overheads. This slide provides a summary overview of their findings. Here I've depicted the 4 different main steps that these authors were looking at. So we can see from the left to the right, we start with DNA extraction, then library prep and sequencing, then we have clinical analysis, which is interpretation, and then lastly we have confirmation and reporting. So I'd like to draw your attention specifically to this clinical analysis, which in this case, you can see that it is quite a processing, a high maintenance process in this case. Firstly, SNVs in known genes would be analyzed. If required, a CNV calling pipeline would be triggered after the first analysis to try and identify CNVs that could be relevant for that patient. For example, CNVs that could be in compound heterozygosity with an SNV in a recessive gene. Then the authors were looking at those CNV variants, and then later on, if nothing was found in known genes, then a research analysis was triggered. So we can see that these include several analyses within this step, which is really, a very human-led and often manual process. So in terms of their findings, obviously what they found, not unexpected, is that sequencing is the most expensive step of the process, accounting for 79% of the total costs. However, clinical analysis is the most time-consuming step and is actually also more costly than expected. So if we look at the concrete times and costs, overall, the paper estimated that the price of a trio sequencing is around £7,000. And if we think about a single genome sample, that works out at around 3,000 US dollars. So pretty, much higher than, the expected $1,000 genome that we all wanted. In these 2 tables at the top, I have shown the associated costs and times of each step of the sequencing pathway. And I think a take-home message is here really is that clinical analysis, even in a simple rare disease case, can take up to 11 hours. So a simple RD case is a case where which can be easily solved by mutations in known genes. Complex RD cases, their clinical analysis can take up to 16 hours of clinical interpretation. And these are cases that require research, gene agnostic, more exploratory analysis. Another interesting take-home message from their results was also that 70% of the analysis costs is actually attributed to staff time, which really, again, further highlights these manual process and expertise that is required for the analysis of these patients. So why does this matter and why should we all be discussing it? Well, this is important because it has clinical implications for the delivery of genomic and genetic services, especially for patients, and also implications for staff. So if we think about a clinical interpretation from 11 to 16 hours, this pretty much means that we can do 1 case per day, which is pretty low case throughput. This means that we'll be increasing the backlog of patients to analyze, and the ability for the lab to cope with turnaround times will be greatly compromised. For the patients, this means that returning results to them will take much longer, and specifically in the case of the US, this has consequences for reimbursement. In terms of implications to the staff, obviously this leads to burden on staff and impact on productivity because you cannot really analyze that many cases that quickly. And ultimately, this leads to a difficulty to scale up, and especially when demand for genetic services are on the rise. So how can we maximize workflow efficiency, diagnostic yield, and confidence using Congenica? So Congenica's clinical decision support platform really sits in between the primary analysis, so the sequencing itself, and the return of a patient diagnosis. So Congenica streamlines and automates the secondary and tertiary analysis to enable the rapid processing and interpretation of patient NGS data. Our platform is secure, collaborative, and highly scalable. And here, scalability, this has definitely been proven with our engagement with Genomics England and now the rollout of the genomic medicine service. And we have support for both current builds of the reference genome, so GRCH37 and GRCH38. And as I said before, we can process whole genomes, whole exomes, and gene panels. So in terms of features in this slide, I'm going to walk you through some of the key features that we have in the platform. This list is not exhaustive. And you're going to have the opportunity to see some of these in action in my live demo. So the first key feature that we have is the ability to call, so pipelines to call, and the ability to interpret multiple variant types in one single platform. Another feature that we have is the ability to automatically generate multi-generational pedigrees for your families that you are analyzing. And these can be very helpful when you're trying to visualize disease segregation within the family, especially if you're looking at a very large pedigree. We also have an integrated genome browser. This helps users to visualize their patient variant in the context of both familial data, so from other family members that are part of the same family, but also in the context of multiple reference data sets, which really helps to contextualize everything that we know about the variant and helping you to more quickly make a classification. We have a suite of quality control information that we generate for VCFs and BAM files and FASTQs. And we also generate gene coverage information to give you an idea of the sequencing performance of individual genes that you are interested in. So all of these QC and sequencing metrics really help you to gain confidence of the variants that you are reporting. And this can be greatly, very important, not only in positive cases, but also when you're issuing negative reports for your patients. And lastly, we have the ability to perform ACMG guidelines classification, and we have fast and flexible patient reporting. So having learned a bit about Congenica, our mission, and some key information about our platform and our features, I'd like now to move on to the next part of the presentation, where I'll be sharing with you real-life cases where workflow efficiency gains were possible thanks to the use of Congenica's clinical decision support platform. So I'll be walking you through the user journey in three different examples. So we'll be looking at the value of three different features, set of features, let's say. So the first one I'm going to show you, the value that can be gained in terms of efficiency of interpreting SNVs and CNVs simultaneously right from the start. In the second example, I'll be showing you how efficiency gains can be attained by instantly prioritizing variants based on the patient's phenotype. And in the last example, I'll be showing you the value of leveraging existing knowledge to focus your interpretation and be able to look at relevant variants more quickly. So if we start with the first example, this will showcase the value of using Congenica's pipelines to call SNVs and CNVs simultaneously, and the ability to perform variant interpretation in both variant types at the same time. So the real-life clinical case that I want to use to demonstrate this value is a 10-year-old boy singleton, so parental information was not available when our user did perform this analysis. And this boy was submitted for exome sequencing on the basis of a referral of disproportionate short stature, vertebrae abnormalities, and fusion of the tarsal bones, which are bones in the foot. So to our user, this was highly suggestive of a very rare type of a skeletal disorder. So if I now jump to Congenica and take you through our user's journey in uncovering these patient diagnoses, before continuing with our story, I just want to give you a very quick overview of our user interface so that we can all be located quite easily in here before I continue. So once you log into Congenica and you're looking at a given patient, every patient in our UI has this split between left tabs and then tabs in the right. So the tabs that we present on a patient basis on the left are tabs that are related to the clinical information and the clinical presentation of the case. So they contain all of the relevant information that users need to know before they can start looking at the genetic variation of the case. So one example of a tab that is related to clinical information is the HPO terms tab, or human phenotype ontology. So this is a tab where we store the phenotypes that have been supplied as part of this case and a part of the processing of this case. And this is where users can store that information and also refine their phenotypes. The tabs on the left, on the other hand, are tabs that are particularly related to the sequencing data. So they can be quality control tabs, for example, that let you know on a sample basis several sequencing metrics, but they can also be gene coverage tabs which calculate on a per gene basis the coverage of your genes of interest. And lastly, that I want to point is the variants tab, which is actually where I'm going to be spending the majority of the time in these demonstrations. The variants tab is where Congenica stores and displays the SNVs and indels, if we are looking at the sequence variants, that have been called in this patient. So if we go back to our story of this first example, and if we replicate the journey that our user took to analyze this case, so our user started by looking at SNVs and indels that were present in these boys' exome. I'd like to draw your attention here to this configuration panel on the left, and these are a set of filters that have been applied by default at the start of the analysis to help the user narrow the focus of analysis into more relevant sets of variants. So our user in this case was using a filter for a virtual gene panel, for example, of a skeletal dysplasia. So just focusing on genes that are related to these expected phenotype, suspected phenotype in the child. Variants that were rare in the population, taking into account multiple reference data sets. And then also variants that were coding and that had relevant protein consequences. So if we start with the rationale that our user took in here, he started by reviewing the variants in this case. We can see that the first two genes are known to have an autosomal recessive mode of inheritance. However, the patient was heterozygous for these variants, meaning that the second hit in this gene has not been identified. So the genetic hypothesis that our user thought was perhaps there is a copy number variant, or CNV, that is in trans or compound heterozygosity with this SNV, and both of them together can cause the phenotype in this child. So that happened not to be the case for the first two variants, but when our user started to analyze the third variant that has been highlighted in these variants tab, and particularly in the FLNB gene, he realized that actually there was a copy number variant that was in trans with this frame shift variant that was here identified. So before showing you how that was done, I just want to point out that FLNB is a gene that is indeed specifically associated with the very specific phenotype that our user was expecting for this child. So if we click on these SNV and review additional information about it, this variant ended up being classified as likely pathogenic. And if we scroll down to our integrated genome browser, we are able to visualize our SNV here in the context of the transcripts, which are displayed at the top, and then in the context of other information. So for example, we have the patient SNV here with a single dot demonstrating heterozygosity, and then several other tracks that I will explain in a minute. So how did our user realize that there was a CNV that was in tran with this variant? So simply what our user need to do was just to zoom out at the level of the gene and be able to quickly spot that there was a CNV that was located between exon 5 and 8 of this gene. It was around a 5 kb deletion. These two variants were later on confirmed to be in compound heterozygosity and present in either parent. So if we continue scrolling down just to look at what other information, in this case, the genome browser is showing us, for example, we can see the SNV in the context of ClinVar variants, for example. We can also see it in the context of NOMAD allele frequency information, for example. And lastly, we can also see in this case whether the CNV overlaps with Decipher CNV pathogenic variants. So these tracks are highly customizable. There are a lot of different tracks that are available, but here I've just given you a flavor of the ones that were looked at in here. So if we just want to get a little bit more details about that CNV in this patient, our user simply needs to go and went to the structural variants tab. And looking at these variants, again, Congenica will let you know that this variant is present in overlaps with other variants that have been termed as pathogenic in Decipher. In terms of copy number, you can learn that is around 1. So in this case, a heterozygous deletion. And then our user also reviewed the base factor, which is the confidence that comes right straight from our CNV calling pipeline, which indicates the probability of the variant being a real event. So giving you confidence that this variant is real. So I hope that these example showcase the value of using Congenica's pipelines to call SNVs and CNVs simultaneously, and also the use of a single integrated platform to perform interpretation of both variant types in parallel at the same time, right from the start. So doing the, and this was highlighted by these diagnoses in this patient of this SNV and CNV compound heterozygote in the FLNB gene. So in terms of benefit, what I've just shown you here really removes the need to run a two-stage bioinformatic analysis, where you first run an SNV calling pipeline, interpret those, and then wait to run a CNV calling pipeline, and therefore reduces the analysis waiting times because your copy number variants are available to be interpreted right from the start. So all in all, this expedites the diagnoses and increases productivity because exactly your samples are not in limbo while you're still triggering other bioinformatic analysis. So if we move to the second example, this one will showcase the value of instantly prioritizing variants based on patients' phenotypes. So this can be done in Congenica via the use of Examizer, which is our variant prioritization algorithm. An exomizer prioritizes variants based on phenotype by taking into account the HPO terms or phenotypes that have been supplied for your patient of interest, and calculating a measure of phenotypic similarity for each gene based on animal models, omnium information, and so on. And then you can use that score to rank variants, as we're going to see in this example. So the clinical real-life case that was consented to be used in this example is a 4-year-old boy. In this case, we're looking at a trio. So both parents were also exome sequenced. And our user chose to use Congenica to analyze this patient and try to uncover the genetic basis of his condition. So the parents were first cousins. So in this consanguineous setting, obviously a first hypothesis would be that a recessive mode of inheritance is happening in this genetic disorder, and the phenotype that the child was reported to have was pontocerebellar hypoplasia, so a progressive atrophy of several parts of the brain, including the cerebellum, so a very severe progressive neurodegenerative disorder. So again, if we go through our user's journey in this genetic diagnosis, and again, we visit the sequence variants tab within Congenica, we'll look at the set of filters that our user was looking at for this case. I'd like to point out here that contrary to the previous example, here our user was really not using a virtual gene panel approach, but rather was conducting a gene agnostic analysis to try and look at the broader potential genetic causes. So again, our user obviously was still looking at rare variants and variants that had relevant coding consequences, but given the clinical information of consanguinity, our user was leveraging our inheritance pipelines to be able to just filter for variants that were consistent with autosomal recessive mode of inheritance, so either homozygous or compound heterozygous. So when our user was presented with this information, and if he was to be sorting variants by default based on maximum allele frequency, which would put your rarer variants or novel variants at the start, the pathogenic variant that ended up being the diagnosis in this case was present at position 35. So the pathogenic variant in this case was a missense variant in TSEN54 that could only be found in page 4 if the user were to be using a maximum allele frequency to prioritize variants. So this is not unexpected because obviously in a recessive setting, and specifically in a consanguineous case, we would expect to see a lot of recessive variants, many of which would be rare in the population. So instead of using the maximum allele frequency, our user chose to leverage the exomizer gene phenoscore that is available in Congenica. So this score gives you a quantitative measure of the phenotypic relevance of your gene to the phenotype of your patient. So once our user did that, TSEN54 missense variant immediately came as top 1 in this patient, helping him to very quickly identify the pathogenic variant without having to review all of the others. So if we look a little bit more closer, here in column 3, we can see the exomizer gene phenoscore for this variant, which was pretty high, so around 0.92. And if we scroll to the right, just to give you a little bit of a flavor of what other annotations we are providing in here, this variant was consistent with the recessive mode of inheritance, as we've seen, was biparentally inherited, so heterozygous in both parents, and was present in ClinVar, but also importantly was picked up by MASTERMIND, because there were quite a lot of papers that actually have cited these variants either in the context of an exact cDNA position or in the context of at the protein level. So if we click in greater detail, just to see what else can be reviewed as part of these variants, we can see the ACMG classification that was used for these variant, and we can review the criteria that were used to call these variant likely pathogenic. And then at the bottom again, we have our integrated genome browser. The difference here from the previous case is that obviously because we're looking at the trio data, we also have the ability to use familial information and display the tracks for the mother and the father in this browser too. And again, I'd like to draw your attention here to MASTERMIND data, which is also available as a track in Congenica. This enables you to quickly review the number of articles and see regionally where other articles nearby your variant of interest have been identified. And then here, via a simple link, you can quickly hook up to MASTERMIND's website and then review the clinical evidence that has been previously published for your variant of interest. If we scroll down, in here we have the ability to see other tracks too. So for example, we can see the protein coding sequence, we can see the orthologs and conservation throughout the species for that amino acid, and then obviously the raw sequencing alignment for that case. So I hope that these examples showcase the value of instantly prioritizing variants based on phenotype via the use of Exemizer. And I think this was clear by these diagnoses of these TSEN54 homozygous variants in this underlying disease in this patient. So why is Exemizer useful? So these really avoids unnecessary review of non-causal variants, helping you to focus in the variants that are more likely to be disease causing in your patient. So this leads to obviously improved speed and ease of analysis, even in complex cases. So even in those cases where you're conducting a gene agnostic approach, Exemizer can be quite powerful. Also, because you have a quantitative measure of the phenotypic relevance of your gene where your variant was found, this also increases your confidence in the right diagnosis that you're making, and overall saves staff time. So the third example of efficiency that I want to show you is the ability to leverage existing knowledge in the form of knowledge bases or your own custom curated variant lists to focus your interpretation in the set of variants that are more likely to be relevant. So the clinical case in here is a little bit different from the others. So here we're looking at, or our user was looking at a couple with 5 pregnancies between 2010 and 2018. And 3 of these pregnancies were affected with a lethal skeletal dysplasia. So the 3 fetuses didn't make to term, and this was a very, very severe, incompatible with life phenotype. So the X-ray here of the first fetus that was conducted by collaborators of our user was suggestive of a very specific type of skeletal dysplasia. And we can see here that there is abnormal skull development, a strong abnormality of the skeleton, and particularly very short and bent limbs, so arms and legs. So since 2010, this couple has been waiting for a genetic diagnosis, and despite several investigations, genetic investigations conducted previously to using Congenica, nothing had really been found. So our user by 2018 just decided to take DNA from these 2018 fetus and really try and analyze it in Congenica to try and understand if there is anything else that can be learned to be underlying this severe phenotype. So if we go through our user's journey again, so in this case, our user was looking at genes via the use of a virtual gene panel, genes that are known to be associated with unexplained skeletal dysplasia. So there's quite a lot of genes, around 300 or so genes that have been associated with different types of skeletal dysplasias. So also similarly as previous cases and as traditionally done in basic filtering approaches, our user was also looking at rare variants that had coding consequences that could be relevant for disease. So after reviewing, there was only 4 variants that survived these traditional filterings. And after reviewing these variants, our user wasn't able to really call any of these variants as potentially linked to these disease phenotype. So here, the genetic hypothesis in this case was quite interesting, and our user thought, okay, perhaps if I don't look at coding variants, there could be actually non-coding or regulatory mutations that could be underlying these phenotype in some shape or form. So because non-coding and regulatory mutations are very difficult to interpret and analyze, as I'm sure you all appreciate, our user thought of using a feature that is available within Congenica, which allows you to instantly look at your set of curated variant lists or knowledge bases, and the user leverage his own customly curated list of Mendelian regulatory mutations. So actually, this was curated by Smedley et al. And our user used that research that has been presented in that paper, which compiled the set of regulatory mutations and non-coding variants that have been linked with Mendelian disorders. So once our user leveraged curated variant list, he immediately spotted 2 non-coding transcript exon variants in the untranslated RMRP gene. So these 2 variants were heterozygous and were later on confirmed to be in compound heterozygosity with each other and were present in the parents. So this gene, RMRP, is not a coding gene, so it encodes an RNA component of an endoribonuclease and is involved in ribosomal processing and has been specifically associated with several types of skeletal dysplasias. So both of these variants, by reviewing the curated information that is stored as part of the curated variant list, both of these variants have previously been reported in compound heterozygosity in other skeletal dysplasia patients, but a lethal phenotype has really never been previously described. So these 2 variants are known to be regulatory enhancers that alter RNA processing and ultimately lead to these severe phenotype. So if our user was not really using curated variant lists, and if he had to resort to the traditional filtering approach, you would have to be looking at ensemble consequences for known coding and potentially regulatory variation. So for example, you would need to be looking at, for example, UTR variants, known coding transcript exon variants, and other types of regulatory variants. So if that is done, as in a traditional filtering approach, it would be, we can immediately see that the user would be presented with many other genes and variants to review, which obviously many of them would be, or vast majority of them would be very challenging to interpret, and many would end up just being obviously variants of uncertain significance. So I hope that these example showcase to you the value of leveraging existing knowledge in the form of curated variant lists, including our own curated variant list, to focus your interpretation and particularly uncover unexpected variants that can underlie a phenotype. And in this case, this was a really remarkable diagnosis for this couple with the identification of non-coding compound heterozygote variants in the untranslated RMRP gene. So in terms of value, leveraging existing knowledge in interpretation in Congenica instantly helps you to detect known variants and particularly unexpected variants, and accelerates your variant interpretation because it removes the need for you to be analyzing a lot of other variants that would just be increasing noise and would be very challenging to interpret. And finally, this increases diagnostic yield in a case that would otherwise be negative. And lastly, it obviously increases your case throughput because you're able to get at your pathogenic variant again much faster. So if we go back to our initial problem and the initial challenges that we discussed as part of this webinar, and we look at the concrete times and costs of clinical analysis, and if we do a like-to-like comparison with Congenica, we believe that Congenica reduces interpretation costs by up to 95% and helps you to get at the answer 20 times faster. So we have estimated these metrics based on around 2,000 Genomics England cases that have been analyzed using Congenica. So many of these cases, actually, I'd like to point out, prior to enrollment to the Genomics England project, they have been through an extensive prior genetic testing and nothing had previously been found. So these can greatly show the value of using our clinical decision support platform. So just to wrap up, I hope that this has been valuable in showing you how our platform can improve your diagnostic yield, helping you to get at the genetic answer faster, but also with a maximized workflow efficiency for your services. And obviously an increase in your diagnostic confidence on the variants that you are classifying and the diagnosis that you are making for your patients. So I presented you three examples that showcase specific features, but there are many other features that can be explored in our platform. So if you've liked what you've seen, feel free to just go to congenica.com/demo and just book a personalized demo with a member of our team. We'd be very happy to take you through our platform. And then obviously if you like that and you would like to evaluate the software and get full access to it and test your own data, you can do so in an evaluation. And this before getting obviously onboarded with full customer support and training sessions. So thank you very much for listening to me today. I hope that this webinar was of value to you. It has been a great pleasure to share with you the examples of real impact and the great work that professionals in the field are doing for patient diagnosis using Congenica. And I hope that it was of value as you as well continue to make your own inroads into patient diagnosis. Thank you very much, and I'll hand over to Mark.
Thank you, Eva. As we switch over here, I'll open up the floor to questions, which I think Kate has been fielding. I'll also echo what Eva said about getting access to Congenica to test out in demo form to begin a trial. Likewise, MASTERMIND comes in two different varieties, including a basic edition, which is available to you at no charge, but comes along with a 14-day or 2-week trial of MASTERMIND Professional, which includes a lot of those features that I alluded to earlier, including phenotypes, drugs, and ACMG classification keyword searches. And you can access either of those, the MASTERMIND or Congenica access points, through the websites that are displayed there. And with that, I'll pass it over to Kate, who I think has a couple of questions that have come in that we have time for Eva and the Congenica team.
Thank you, Mark. And just to echo that, I will be sharing the links to MASTERMIND and Congenica in our follow-up email. And we do have a couple of questions for Eva. In terms of curated variant lists, how many do you reference? How can we access them with your software? And can we search for variants in each one?
Thank you, Kate. Yeah, that's a really great question. So at Congenica, in our platform, we support multiple curated variant lists. So particularly and including some of which I presented in these as part of this demo. So for example, we have support for ClinVar, Decipher, more recently MASTERMIND data 2, and then in terms of CNVs, for example, I can give you an example of DGV and also ISCA for larger structural variants. Importantly, our users can also create their own customizable curated variant lists, and they can do so very easily via a curated variant list feature, where they can import their own data and start to curate as well their variants and add variants to those variant lists once they finish analysis in the cases. The users also have the functionality to store several types of information against each entry in those curated variant lists.
Okay, great. And I think we have time for one more. How are gene panels managed within your software? Do you provide your own panels, or can a user make their own gene panels?
Thank you, Kate. So we have Congenica provides certain gene panels that are available for use for users. However, again, users have flexibility to create their own gene panels. And these can be done via a gene panel curation, gene panel management tool, let's say, where users can import lists of gene panels, particularly if they're very large. That's very easy to do within Congenica, and users can store quite a lot of information about those genes. For example, they can select which mode of inheritance is known to be relevant for each gene, and for example, select a preferred transcript in the analysis when that gene panel is applied. They can select a specific gene panel, a specific transcript, sorry, for each gene. So they can also store multiple versions of the same gene panel. So they pretty much have complete flexibility to edit, export, delete, and modify gene panels within the system.
All right, thank you. So I just wanted to say thank you to everyone for submitting your questions. I'll be sending up a follow-up email with a link to this recording and the slides, and then we'll also have the questions and answers on our webpage. So if we didn't get to your question, it will be posted there. Just want to say we appreciate you taking the time to complete a short survey that'll come at the conclusion of this webinar. Your feedback is very important, and it helps us plan for future webinars. Please be sure to visit genomenon.com or congenica.com to learn more about us, and follow us all on social media at Genomenon or at Congenica. Have a great day, everyone.